We're going to take a look at biofilms and quorum sensing. So we've talked a little bit about biofilms already, but remember that biofilms will form on surfaces and the microorganisms will cover themselves with this slimy layer. And that consists of a extrapolymeric substance that's made of polysaccharides, proteins, and water. And the microorganisms like to live in the biofilms because it forms a nice protective surface that protects them from the environment and then also from our immune cells and also from antibiotics as well. And the other form that the microorganisms can live in the environment would be a planktonic form. So that's a kind of a free living form that is not in the biofilm. And the medical importance of the biofilms is that they're hard to treat. So remember we talked about these biofilms getting on medical devices, so things like total hips, total knee replacements, and they can also get on urinary catheters, IV catheters, and once they get on those devices, you just have to take them out and then replace them and start over. And so this is from a total hip replacement. They've cut open the joint capsule and you can see all the pus that's in there. And so they would take the total hip replacement device out and then they actually would put that patient on antibiotics probably for about 6 weeks. And then once the infection is cleared, then they would go back in and replace the medical hardware and then the patient would be able to walk again. So it can be a long, painful process because the patient has to go through 2 surgeries to replace all that. Replacing a urinary catheter or an IV catheter is much easier. And then in dentistry, we also have problems with biofilms as well, because they form the plaque on the teeth, and if you're not careful in brushing all of that off, that can irritate the gums and cause gingivitis. And if that is not taken care of, the gingivitis can then lead to periodontal disease and tooth loss. And this just shows the formation of biofilms. So you can see these planktonic bacteria that are just swimming around in the environment. They will attach to the surface, start secreting the slimy layer, form the biofilm, and then later on the parts of the biofilm could break off and go attach elsewhere as well. Okay, and this is what the biofilms look like under an electron microscope on the top left. And you can see how the bacterial cells are very well protected and these little nests in there inside the biofilm. They're surrounded by the slime layer. And that makes it very difficult for our immune cells and antibiotics to penetrate that and actually kill off the bacteria that are living inside of that biofilm. And you can see that the conditions inside the biofilm vary depending on where the bacterial cell is living. Inside the biofilm, deeper inside there, there's not as much food and lower oxygen levels, so you're going to have different species of bacteria living inside, deeper inside the biofilm than you would on the surface where there's more food and more oxygen. Okay, and then the next thing would be quorum sensing. And this would be important to know, so I would put a big star on quorum sensing in your notes. And this is particularly interesting because quorum sensing is talking about individual bacterial cells talking to each other and actually coordinating their behavior to carry out, some activity that they want to do when a certain number of bacterial cells are reached. So let's just take a look at a picture of quorum sensing. Oh, and then before we get to that, the reason it got its name is if you've ever been in a club, you have to have a quorum of members, a certain number of members, before, club business or a vote could, take place. And so that's where the name came from. Okay, so here we've got on the left, we've got a bacterial cell. And our bacterial cell is producing some signaling molecules. Okay, that's what these little silver balls are. And as the bacteria divide and produce more bacterial cells, each of these bacterial cells will then start secreting these signaling molecules into the environment. And so the signaling molecules start to build up in the environment. And then we get this concentration gradient. And so now you've got a high amount of these signaling molecules outside the bacterial cells. And so now they start moving back into the bacterial cells. And now these signaling molecules will bind to the DNA and turn on certain genes. And that will cause this population of bacterial cells to do certain things. Okay, and so this is how the bacteria sense that, oh, we've got this many cells now, and now let's go ahead and turn on, these genes, and maybe, for example, we've got Pseudomonas aeruginosa, and they say, well, we need to get to this certain number of bacteria, and once we do, now we can go ahead and make a biofilm. Okay, and so they'll send these signaling molecules out, and once they sense, oh yeah, we've got enough bacteria, let's go ahead and make a biofilm. And Pseudomonas will also be sending out signaling molecules that will sense that, oh, we have a certain number of bacteria, let's turn on some genes and make some toxins and make this person really sick. Okay, because what they do is they kind of fly under the radar and avoid attracting the attention of the immune system until they know that they've got a big enough number of bacteria. And then they're like, oh, now we've got enough bacteria that we can overwhelm the immune system. Let's go ahead and turn on this gene to make this toxin. And now we can make this person really sick. And, the immune system can come after us, but we've got enough bacteria now that we can, make this person sick. So that's quorum sensing. So it's really interesting that these bacteria are able to communicate with each other and actually coordinate their behavior and turn on genes, and then they all do the same thing at once. So that's quorum sensing.